Hey guys, welcome back to a new video by Biology with Zangshan. So the June 2023 is over and today we have a paper that we can discuss, which is the 0610/41 May-June 2023. This is the paper 4 theory extended paper. So the link of this paper will be in the description below and make sure you check it out. So let's begin with the first question. Question 1a. Figure 1.1 is a diagram of a digestive system. So they just give you the entire alimentary canal, which this is located in chapter 7. Each letter may be used once, more than once, or not at all. State the letter of the part shown in figure 1.1. So you need to find which organ actually produces bile, which is the liver, which is K. Okay, so the answer is K. And which organ that produce gastric juice, which is the stomach, which is C. And then that produces urea would be the liver again, which is K. I'll be using because it say it can be more than once. Okay, and where maltose is digested is at the lining of the epithelium, which is located in the small intestine, and this is located in H. And where trypsin acts is in the duodenum or the ileum, which is located in the small intestine also. So the answer is H again. So your answer should be K, C, K, H, H. 1 mark is worth for 1 mark out of the 5. Okay. Question 1b. A student investigated the effect of bile on the digestion of fat in milk. They set up three different test tubes. Test tube A contained milk and bile. Test tube B contained milk and lipase. Test tube C contained milk, lipase, and bile. They used an indicator that is a pink in alkaline solution, colorless in acidic solution. They added the same volume of indicator to each test tube. The student observed and recorded the color of the contents of each test tube at 0 minutes, 20 minutes, and 40 minutes. Table 1.1 shows the results of the investigation. So you can see pink, pink, pink. Pink is meaning that it's in alkaline. It means it has remained alkaline. And if it's colorless, it's become acidic. Okay, so they want you to explain the results for test tube B and C. So what I can say is that the pH is decreasing, meaning it goes from a alkalinity phase to a more acidic phase. So where it becomes more acidic due to fatty acids being breaking down in the process, which affecting the pH. So when the fatty acids are broken down, the pH will also lowers. Okay, in test tube C, the production of fatty acids is quicker because in 20 minutes time it has already turned colorless, which is quicker than test tube C than test tube B, because in test tube B it only turns colorless after 40 minutes, which definitely allows the bile to emulsify fats. Okay, so that's how you get your 4 marks. It's just explaining the results in the table. Okay, explain the purpose of test tube A in table 1.1. So you can see, right, in test tube A, it's pink, 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 means it has remained the same all the while. So you can see that test tube A contains milk and bile, no digestive enzymes here. So what does this do is that it acts as a control. Okay, what's the control mean is to show that the independent variable is not actually affecting the main point of the experiment. So you can see that the bile does not break down the fats into milk because the fats is still one big molecule. It has to be broken down into a smaller molecule in order bile can be emulsified successfully. So the answer is this. Okay, question 1c. The action of lipase is affected by temperature. Figure 1.2 shows the axis for a graph of the effect of temperature on the activity of lipase. Complete the graph by drawing a line to show the expected effect of temperature and on the activity of lipase. Drawing and adding a label line and a label to show the point at which all the lipase has been denatured. So what's this? They want you to draw an enzyme graph which is the temperature is being affected here. So what does this do is that it always increases and it has an optimum pH here, and it starts to go down and denatures at a higher temperature. So you can see that temperature is here and enzyme activity. So as it goes, as the temperature increases in the beginning, the enzyme activity is higher, and once it passes the optimum pH, the active site starts to change, and eventually it all denatures. So most people, they will get 1 mark only, but you must remember that you have to add a label line, which is this point where it touches the x-axis. Then once it touches here, label it, draw an arrowhead, all enzymes denatured. That's how you get your 2 marks here. Okay. Question 1D. Explain why lipase cannot be used to catalyze the breakdown of proteins. What does it mean? It's just that says that why lipase cannot break down proteins. Okay, so you must say that the substrate only allows fat molecules to bind. Okay, the shape of the active site is not complementary to protein, but fats only. Therefore, no enzyme substrate reaction would occur. So this whole point of my keywords using complementary, active site, the shape of it, and the substrate is only able to allow the fat molecules. It's all about this word specificity. So the whole point of this is that this answer, you must talk about how the substrate is going to bind with the active site and how is it complementary to allow a reaction to happen, which is the 3 marks. Okay. Question 2A. A student investigated osmosis in potato plant cells. Okay, he immersed cubes of potato tissues in water and different concentration of sucrose solution for 30 minutes. The masses of the potato cubes were measured before and after immersion. The percentage change in mass were calculated. So here's the table. You have to see, they ask you to calculate the percentage change in mass. So using the information in table 2.1, calculate the percentage change in mass at 1.00 mole per dm minus 3. So give your answer in 2 decimal places and show your working, of course. So they want to find percentage change. So when this happens, right, percentage change can be either a positive change or a negative change. So if this case, we use the mass of potato cube after immersion over before and times it by 100. But before that, we need to find what is the difference of it. So this is the formula that we are using: after minus before over before times 100. So 1.11 minus 1.28, you get a negative value. Okay, this is 1 mark. Subbing into the formula, minus 0.17, this is your answer. And over the before value, 1.28 times 100%, you get minus 13.28125. Give at least a few significant figures behind, and then they ask you for a 2 decimal place, which is 28 minus 13.28. So the answer is minus 13.28. That's how you get your 3 marks. Break down all one by one, step by step. Okay. Question 2II. Using the information in table 2.1, explain the differences in results between the 0.6 mole dm and the 0.8 mole dm sucrose solutions. Use the term water potential in your answer. So this is definitely a link to osmosis explanation in here. So you can see the 0.8 and the 0.6, there is a much larger change in percentage. So you can see that the 0.8 mole dm potato cubes loses a greater mass due to water movement of water out of the cubes by the process of osmosis. So that's where you get your first mark here, is that it loses at a greater mass, because look at the mass they are losing is at minus 8.2, and for the 0.6 potato cubes, they only lose minus 3.01. So there's a possibility that the water has left the potato cubes and the water moves from a higher to a lower water potential. So this is the osmosis explanation. They want you to use the term water potential. That's the link. And the sucrose solution have a lower water potential than water. Okay, because what in osmosis, water is easier to move in and out than a sucrose solution, which is probably too heavy and the molecules couldn't penetrate through the partially permeable membrane. So this makes the 0.8 mole dm potato cube having a greater water potential in the end. Okay, that's how you get your 5 marks. Alright. Question 2 III. Describe the expected appearance of a cell from a potato cube that has been immersed in distilled water for 30 minutes. So this is quite simple. So when you immerse a potato, just a potato stick inside there, it's a plant cell, right? So what happens is that it becomes more turgid because water will move in from a higher water potential to a lower water potential, and therefore making the vacuoles inside the plant cell bigger and larger, and it's expanded, of course. Okay, that's how you get your 2 marks. Question 2b. Describe how the process of active transport differs from the process of osmosis. So this is basically what is active transport and what is the difference between it. So active transport goes against the concentration gradient and it uses energy from respiration. At times, it also involves the use of carrier proteins. So this is also one important thing that we have to know, is that active transport, they not only just uses energy from respiration, but they also use carrier proteins, because proteins are mostly involved inside all cells' daily movements. Like their possibly features, because 50% of the dry mass of any cell is made out of protein. So that is your answer. Okay, question 2c. State the type of plant cells that use in active transport to help absorb mineral ions from the environment. Root hair cells. Okay. Question 2d. Explain the effect of a lack of magnesium ion on the color of the plant leaves. So this is the mineral requirements. So magnesium ions help to produce chlorophyll. If there is a lack of magnesium, the plant's leaves will turn yellow because there's no more green pigment inside the chloroplast anymore. Okay. Alright, question 3a, figure 3.1 is a photomicrograph of some cells lining the trachea. Okay, so they give you a label X and a goblet cell. So describe the role of goblet cells. So the role of the goblet cells helps to produce mucus and also helping to trap dust and pathogens. 2 marks. Question 3, part 2, explain how cell labeled X is figure 3.1 is adapted for its function. So the X here is the ciliated cells. So the X is the ciliated cells and helps to beat back and forth to move mucus away. Okay. Question 3, part 3, state the name of one other part of the body where the type of cell labeled X in figure 3.1 is found. So they're basically saying that in ciliated cells, where can you also find ciliated cells? So other than the trachea, you can see this in the bronchioles, the oviduct. You know why oviduct is able? Is to allow the movement of the egg to move back and forth. So the egg is only allowed to move by this peristalsis movement of the ciliated cells, which helps to beat back and forth. Okay, question 3b, table 3.1 contains some features of the breathing system. So they want you to see this table 3.1 to show what actions of each features to help cause inspiration, which is breathing in. So the diaphragm during inspiration will contract and the external intercostal muscles will also contract. At the same time, the pressure in the thorax will however decrease and the ribs will move up and outwards and the volume will increase as you allow more volume of oxygen entering into the lungs. Okay, state the name of the gas that's excreted by the breathing system. It's very easy, it's carbon dioxide. Question 3, part D. Good ventilation is one feature of gas exchange surfaces. State 2 other features. So it could be thin surface, it could be wide because it's one cell thick, there's good blood supply, whereas a thinner diffusion distance, and there's of course a larger surface area for more breathing and more successful breathing to help deal with oxygen depth if you're doing exercising. Okay. Question 3, part E. State the name of the gas exchange surface in humans, which is the alveoli or the alveolus. Okay. Question 4a. Figure 4.1 shows the effect of light intensity on the rate of photosynthesis at different temperatures and concentrations of carbon dioxide. You can see that there's a difference in the 0.4% and the 0.04% in the CO2 concentration, and there's a maintained temperature here. So they want you to, for this question, they want to ask you to describe and explain the reasons for the shape of lines in B and C. So you can see, right, they both level off at the same time, but just that there is a significant difference. You must say why it happens like this. So how do you answer this question is you have to break down what is a similarity first, then only you go to the what makes it different. So for both lines B and C, the light intensity increases, which allows the rate of photosynthesis to increase. Then it starts to remain constant and levels off. This is because light provides energy for photosynthesis. You can see that I further explain this part. Why? Because they want to ask you to explain the reasons for this happening. And also in both B and C, the line where light intensity limiting, line C have a lower rate of photosynthesis due to a 0.04% carbon dioxide concentration instead of a 0.4%. So you can see that I'm using a comparison between the difference of the carbon dioxide concentration, and this makes carbon dioxide a limiting factor for line C and growing and giving the higher rate of photosynthesis and a higher line for line B. Okay, so this is how you technically explain. Always start off to see what is similar. You can also say the temperature is the same and doesn't really change. It's more of just the carbon dioxide concentration limiting this process, because carbon dioxide is needed for as the reactants for photosynthesis. Okay, let's move on. Question 4b, okay. C6H12O6, which is technically glucose, is one of the products of photosynthesis. State the chemical formula of the other product, oxygen. But you cannot write oxygen. They want chemical formula, which is O2. Okay? Alright. Outline how carbohydrates are made during photosynthesis are used in plants. So technically they are referring to carbohydrates as glucose or sucrose or cellulose. They are carbohydrates made in plants. So the glucose made from the products of photosynthesis is needed for respiration to release energy. Glucose is also converted into sucrose and to be stored into the roots as zinc through the phloem. Okay, some glucose is used to build cellulose cell wall to maintain the plant structure. So this is how you expand your answers. So you must say that what are the uses of basically the sugars inside plants, what are they used for to make sure that the plants are doing well and healthy to maintain its structure. Okay, question 5a, this is protein synthesis. So question 5a, part 1, state the name of the parts labeled X, Y, and Z. So X, Y, and Z. You must be familiar of the steps of the protein synthesis and you should know what is going on. So X is the nucleus. Why? Because the DNA is located inside. And Y is the mRNA, because we know that it sends out mRNA to the cytoplasm. And Z, where the mRNA passes through the ribosomes in one of the stages, and therefore Z is the ribosomes. Okay, state what determines the sequence of amino acids in the protein that is produced, the sequence of bases in the DNA. So this is the answer. Okay, explain why the sequence of amino acids is important in the production of receptor molecules for neurotransmitters. So this is a bit tricky. They're asking you why DNA is important in the production of receptors molecules in neurotransmitters. So the sequence of amino acid, it determines the shape and the active site of the protein when it's produced, of course. So where a complementary shape is definitely required for at least a successful binding to the protein receptors in the other side when it passes through the synapse with the neurotransmitters. Okay, so it has to be the correct one so that successful binding can occur. Okay, explain why body cells have different specialized functions even though they contain the same genes. Okay, why is this happening? It's because not all genes are expressed. Not everything is being used up. maybe, okay, for example, a particular like breeding, for example, they don't need everything just to initiate a breeding process. So only some are actually shown. Not everything is given out. So and cells only produce when the body needs. So when the body has already excess of it, you do not want it again. So you just need those that who are basically what the body really needs. You only produce out, and definitely they are not going to show all its features. It's only some of them coming out and express them. Okay. Part C, allele frequency in a population can be changed by natural selection and artificial selection. State the meaning of the term allele. So allele means an alternative version of a gene or another form, new version of a gene. Okay. Part D, describe how artificial selection differs from natural selection. So just saying that what's the difference of artificial selection comparing with natural selection. So artificial selection is the process of humans involve choosing desired features from animals and mating them together for a generation to come. This is to breed so that they can survive. And technically artificial selection is a much faster process than natural selection, because natural selection takes a long time just to get the features. And however, this can decrease the fitness, decrease the biodiversity of these animals being artificially selected. Okay. Part E. Mutation causes formations of new alleles, which increases genetic variation. State two other sources of genetic variation in population. Okay, this question, right, I know there will be a trick. We all will fall that we wanted the causes of mutation, meaning like nuclear, all those that affects the genetic variation. But this time they are not asking for this one. They are asking for two other sources of genetic variation in population. So it means that it's not going to be affected by anything that caused by humans. It's more of just generally and naturally how this is gonna happen. So meiosis, why meiosis takes part in this, because diploid to haploid, and this becomes again, they can mate, and this causes a random mating. It's not forced to be mated. This is random mating and random fertilization. So when this happens, this gives genetic variation. Why? Because you're not producing the same thing. You're producing different kind of humans. So different kind of humans, they're producing different kind of genetic variation. So the answer is meiosis and random mating or random fertilization. Okay. Question 6a. A scientist monitored the changes in the pH in muscles before, during, and after 2 minutes of vigorous exercise. So the changes of pH are caused by the production of lactic acid. So you just basically have to see what's the graph happening, and they ask you to complete the sentence, just like fill in the blanks. So the pH decreases from, if you can see, it's about 7.07. So from 7.07, it has decreased to at least 6.55. Okay, so when this happens, you write down it decreases from 7.07 to 6.55. So there is not enough oxygen supplied to the muscles, therefore the body will respire anaerobically, and the lactic acid produced builds up in the muscle, causing an oxygen dam. It takes about 33 to 34 minutes. So you can see from here to there, it takes about 33 to 34 minutes to go back to that initial level that we see. So if you can see, I pull out the line, it's about here, which takes about at least 33 to 34 minutes in estimate, and the pH value increases after vigorous exercise has ended as lactic acid is transported from the blood, or you can say the muscles probably, and it will be transported to the liver for breakdown. And during this time, the breathing rate or the heart and pulse rate will remain high. So this is the 6 marks. Okay, last part. Yeast can respire anaerobically. Complete the balanced chemical equation for the anaerobic respiration in yeast. So C6H12O6 will become 2C2H5OH. This is basically ethanol or alcohol, and carbon dioxide will be released in the process. Okay, and yeast belongs to the kingdom fungus. Okay, state one cell component that is present in yeast cells but is absent in animal cells. Cell wall. Well, because fungus and plant cells are almost the same. So the difference between yeast and animal is that yeast has cell wall and animal doesn't have cell wall. Okay, so that is all for this paper. So I hope you understand. If there's any doubts or anything that you feel unsure, please make sure you write down in the comments below. I will try to answer them and get it sorted. So I hope to release more past year papers in the future. So thank you for watching, guys. See you in the next video. Bye bye.